Hi guys, and welcome to Biological Sciences for Year 10. This is Activity 6C, where we're going to just give you some extra hints for solving some of the harder problems that you might face in Activity 6B. Okay, so one of the things that you may come across is you're given ratios. So it may say in a biological test, a whole range of plants were crossed and there was a certain number of different types which were generated. So for example, in this case here, you could have a question which said there were 29 yellows and 8 green plants which were generated. And from that you'd have to work out what the genotype of the parents was likely to be. So what you would do there is look at the ratio. You would convert them into nice round numbers. Now the numbers don't have to be exact. They're just a rough guide, because remember we're talking about probability here. But basically, if you take this situation here in example 1, you've got 29 and 8. Well, if I can round them up basically to 30 and 10, which is a 3-to-1 ratio. So basically what you can work on there is, if you went back to each of the 3 examples that I showed you in Activity 6A, you would be able to work out that basically if you had a homozygous dominant with a homozygous recessive, you're going to get heterozygous and they're all going to be the same colour. Well, that wouldn't work. If you then took one where you've got heterozygous with heterozygous, what we found was we would get a situation where we would get 3 would be one colour and then the 4th would be a different colour, so you get a 3-to-1 ratio. So in that case what you then can state is that your parents have to be heterozygous for that specific type of trait. And obviously if we then had a heterozygous with recessive, remember we had 50-50, 50 yellow with 50 green. So basically this allows you to work, you have to work backwards, and this is why it's slightly harder, because you have to sort of interpret what the data is telling you, convert it into a ratio, and then look at each of the 3 Punnett squares and say, that ratio fits with that one over here or that one there. So as a result, the most likely probability of the genotypes of the parents is this and this, and then you would explain it. So this is pushing into that sort of A grade sort of question. So that's one area that you can work on. Second area you can work on is you might be given an offspring and a genotype of the offspring, and then expected to sort of explain what are the parents. So in this case you have to work backwards. So if we've got an offspring which is little b, and basically we've got the letters corresponding to big B being brown and little b being blue, and little b is recessive, then in order for the offspring to be little b, we've got to work backwards. So where would you get across where you would get little b, and basically you would then have to explain it. Now you can get it from a situation where you've got heterozygous. The gametes could be heterozygous, and as a result, as we've gone through in example 3, 2 heterozygotes linked together will produce a homozygous recessive. You could have a heterozygous with a homozygous recessive, and that way you would have to explain which one it is. So you can see there you would need to be able to state what's happening. Now more often than not you might know what the phenotype of the parents is likely to be, and if you know that the phenotype of the parents are, in this case, brown and brown, you would definitely have to be able to state that you've got basically 2 heterozygous there. You couldn't have it the other way around. You couldn't have a parent being if the parent was homozygous recessive, they would be blue. They'd have a phenotype of blue, so you'd already know that you must have heterozygous linked with homozygous recessive, because obviously the blue has to come out as a double recessive. So you can work it through that way. Now it's a bit confusing, and you may need to go through a couple of examples there, but you need to work backwards and sort of say, well, what would I get if I had these parents go through, do you get your homozygous recessive? The answer is no. Put a line through it, start again, and then explain it in your example. So it's not a case of you being told you've got this and this, homozygous, heterozygous, whatever, and then just run through. That's the basic setups. This is working from your offspring backwards. So again, it's a lot more complicated. Okay, so let's look at the last one that we can look at, and this is what we call a pedigree chart. And in a pedigree chart, what it's showing is the ancestry of various individuals as you go through from one cross to another. So in this case, you'd be asked what is the possible genotypes of box 1 and box 2. This is very, very simplistic, but you'd be given a load of information and you'd have two mates and you'd have maybe the phenotypes, or you might be given one genotype and then expected to go through and say, well, this would have to be this in order to produce this, and this would then move on, etc., etc. And you just follow it through logically. So in this case, what we do is we take the top two parents and we say, look, on the left-hand side, we've got like a homozygous dominant, so capital T joined with a homozygous recessive, little t. Now in this case, we're going to get all of the offspring being capital T, little t, so we get a heterozygous. So that's what your genotype for 1 would be. And obviously the same is going to be the same for 2. Now when you put 1 and 2 together, would they actually produce an organism that's going to be homozygous dominant? And the answer is yes, because you've got 2 T's in the gametes, so those 2 gametes can come together and actually formulate that one. So that's a way of checking. So again, this is a way that you can use this pedigree chart to actually go through and say, oh, this join with this and this join with this, etc., etc. Okay, so I hope you found that useful, and I would suggest you get on with the harder questions in activity 6B. Thank you for watching.